Drug metabolism broad principles. Metabolism increases the excretability of a compound. Broadly speaking, there's two groups, so phase 1, phase 2. Most compounds move from phase 1 to phase 2, but that's not the case for all. Within phase 1, there's a modification of the drug or compound which can result in activation or inactivation of that compound. So a drug can be ingested as an inactive compound, undergo phase 1 metabolism and become activated. This is known as a prodrug, for example ramipril. Within phase 1 metabolism there's three broad reactions, so oxidation, reduction and hydrolysis. Oxidation involves the CYP450s, monoamine oxidase and alcohol dehydrogenase. Reduction involves CYP450s and hydrolysis involves esterases and imidases. So once drugs have undergone phase 1 metabolism, move on to phase 2. This adds a group bond which is water soluble, making the drug more inactive and excretable within the body. These reactions include methylation, so you add a methyl group onto the drug, glucuronidation, acetylation, sulfation, or even an amino acid, so like glycine. It's important to remember that metabolism is in the body in order to produce normal metabolites for endogenous processes. Therefore, the liver is the most important site, but metabolism occurs in a range of different sites. These include extrahepatic sites. The CYP450s, obviously the most important phase 1 metabolism pathways, and these are in a range of different places: kidneys, brain, adrenals, GI tract, lungs and placenta. But within the lungs is the ACE, within nerves is monoamine oxidase, catechol-methyl transferase, metabolism of adrenaline, noradrenaline. Within the plasma there's red cell esterases, Hoffman degradation for urticaria metabolism, and within the adrenal gland there's a range of different steroid, essential steroid metabolic pathways. Cytochrome P450s, this is a massive subgroup involved in phase 1 metabolism, 50 enzymes, and only 6 of which are involved in 90% of drugs. The most important are 3A4 and 2D6. These are involved in normal synthetic pathways for cholesterol, steroids and prostacyclins. They absorb light maximum of 450 nanometers, hence the name. They are located in the liver, but also extrahepatic sites, as discussed. Modification, so we can increase the concentration of these enzymes by giving certain drugs, for example carbamazepine, and we can decrease them by giving additional drugs, for example isoniazid. Within anaesthetics, important enzymes for propofol, benzos, ketamine and fentanyl is 3A4, and volatiles is 2E1.